If not,
Perfect.
let's thank
Thank
Dr.
you.
Pauly once again. Our next speaker today is Dr. Sindhuja Sankaran. Dr. Sankaran is an assistant professor in the Department of Biological System Engineering at the University of Washington State University. Something's wrong. Her research emphasis has been in agricultural automation engineering area since 2013. She is currently leading the phenomic aspects of a couple of ongoing NIFA AFRI grants, where her focus is on developing high-throughput phenotyping tools for field and post-harvest crop trait evaluation in cereal, legume, and specialty crops. Dr. Sankaran holds a BS in zoology, an MS in environmental science, an MS in environmental engineering, and a PhD in agricultural biosystems engineering. Please join me in welcoming Dr. Sindhuja Sankaran.
Thank you. Mic is on. Can you guys hear me? Yeah, I guess. Thank you. So thank you for the student organizing committee and the sponsors for inviting me to be here to talk about phenomics. My background is ag engineering, so I come in the technological side of the things, which is very different from breeding, but I'm learning a lot of things every time I'm participating in a symposium or an event. So today I'm going to talk about sensors and phenomics. Basically, I wanted it to be an educational aspect. So I'm going to talk about small things that's important for the throughput, automated image processing, as well as the benefits and limitations of the machine learning approaches. What does high throughput phenotyping mean? So we heard from Dr. Duke Pauly, and a couple years back, we met at a conference that was basically to figure out what is high throughput phenotyping. And depending on whom you talk to, the definition of high throughput phenotyping can be very different, because sometimes they are focusing on advanced techniques like X-ray CT, which is highly accurate, but probably the throughput is not there. Sometimes it can mean throughput, sometimes it can mean lower cost and rapid, sometimes it can mean automated processing of images, and sometimes it could be a combination of few things. So high throughput phenotyping, for me, it could mean more accuracy, better accuracy, and breeders are okay with relatively lower accuracy but high throughput. So because it's the relative comparison between varieties rather than absolute sometimes. So this is the overall goal for high throughput phenotyping, to figure out how do we evaluate the phenotypes or the traits. I come from Washington State, and we have around 300 different agricultural commodities. You would see there are some animal-based products like milk or aquaculture, but mostly it's plant-based. And we are top ranked in the nation for at least 10 plus crops. I'm fortunate to work on multiple crops that deals with apples, grapes, as well as a lot of legumes and wheat. In terms of breeding programs, as far as I know, there are at least 15 different breeding programs in Washington State, and it is not just the researchers in Washington State University, but also the USDA ARS scientists. So there are centers in Washington State University, and several of those centers have also USDA ARS units and we work collaboratively on many different things. So we do have cereal grains, wheat, barley, quinoa, to name a few, legumes, chickpea, dry beans, dry peas, lentils, horticulture crops, hops, potatoes, camelina, apple, pears, cherries, and many more, including Christmas trees. And many of these breeding programs are supported by the Commodity Commission. So without their funding, their support, their involvement, the success of these breeding programs would not be possible. As far as in our lab goes, we work on several different types of sensors, which range from handheld drones to satellites. So we have the sophisticated handheld sensors that is capable of sensing visible near-infrared spectra from the leaves. We use tractors. Here is one where the sensors are mounted on a tractor. We have different system for tree crops because we want to be able to adjust the height of the sensor data capture much more than this one. And of course, the most exciting and advanced technology, which is drones, and it is growing. It's an growing era. We work on many different types of sensors. We have simple multispectral cameras with near-infrared red edge bands capable of generating generic vegetation indices like NDVI or normalized difference vegetation index is one. We have visible near-infrared spectroradiometer capable of capturing 350 to 2500 nanometer. It's a state-of-the-art equipment, highly accurate. We have an imaging system corresponding to that. We have a Viz NIR, the best possible in terms of accuracy as well as the range. We have 2D LiDAR systems that are highly accurate, centimeter level accuracies, and as well as 3D systems that's lightweight, mainly for drone applications. We use quite a bit of X-ray CT system. We have a wet scope. We utilize their sensors for our applications. Simple RGB, GoPro. In addition, we also have microscopic RGB imaging, and this we use for specialized things. Sometimes we deal with very tiny seeds or tiny crops, and we find this microscopic imaging is useful in identifying the differences. And the thermal cameras. We have different versions. The latest one is the one which has both RGB and thermal integrated together. In addition to that, in our lab, we developed our own platforms. This was a platform that was inbuilt in our lab, and this was a part of the students' master thesis. The advantage of such system is it can be easily adapted. Commercial systems, if you're going to change it, you're going to void your warranty. Here you have the flexibility to add a sensor, remove the sensor, and the most important aspect, the image processing side, which is pretty rapid. In 2 minutes, it can process all the images collected during a series of 10-15 days, and it spits out the phenotype. And we have modulated systems, so each of these modules control a particular sensor which can be adapted. Each sensor comes with a software to operate that particular sensor. When you are using multiple sensors, you should be able to adapt that one software for controlling all different types of sensors. With that, I would just like to say here, all we see is in the visible range, which is 400 to 750 nanometer. But there is a range beyond visible, near-infrared, X-ray, and much more that can be utilized for phenotyping applications. In our lab, we work on many different crops and with close collaboration with several breeders. And throughout this presentation, I'm going to give you several examples of it. Yield potential is important for both legumes as well as grain crops. In tree fruits, including apples and grapes, sometimes we work on disease resistance. We work on water use efficiency, again for grapes, potatoes, quinoa, water use efficiency, and these are just to name a few traits that our research lab works on. Here is an example where we are just looking at the flowering, days to flowering, and this is camelina, which is an alternative crop, and that's a seed sizing that we also do. We not just in terms of seed properties contributing to the quality of the crop, but also seed properties that influence the plant properties, better emergence, spring stand, and other things like that. The second image here is an example of water use efficiency in grapes, which is basically a normalized canopy temperature. And you can see there is no treatment effects. In 2016 winter, we had quite a bit of snow. So even if we had a water treatment, that did not play a role in inducing a stress. Although the excellent thing is still we get in relation to ship with stomatal conductance, we get excellent correlation. And that's mainly possible because we normalize all our temperature and visible near infrared data. An example of tuber, most of our potatoes goes to chip industry. So potato tuber size is an important trait that we do evaluate. And it's possible. We are currently in a stage where we are developing an app to be able to do the shape and size evaluation. Again, another example of a disease tolerance. So we had this AFRI Food Security Grant, which is a $3 million grant, which the title is over here, which our main goal was to help the students, graduate students, in several different types of breeding programs to train them to be able to use the phenomics tools in their breeding objective. So we work quite a bit. So we usually have engineering students paired with plant science students working together, understanding each other. And that process is very important if we really want to develop a high throughput phenotyping technique. So we are 3 cores. We have a phenomic core led by my group, genomics as well as bioinformatics. These are the breeders, including apple breeder, Dr. Kate Evans, and a range of crops. Each crop is very different in terms of cropping systems. Their goals are different. The way they grow is different. So we are learning a lot through this project. So now I'm going to talk about some basic steps of image processing before we move to the machine learning aspect. So as a part of this project, we are, just to start with, we are helping the plant science students, breeding students, basically use the NDVI cameras and extract some basic features. One is a canopy cover, the other is canopy vigor, which can be acquired with NDVI. And the major aspect over here is the throughput rather than innovation in this aspect. We are able to process 25,000 images in 2 days, which is pretty good. We have optimized all these algorithms for multiple crops, and we are able to achieve that throughput. The second example over here is an X-ray CT. So here we are using X-ray CT. In apples, there is a disorder called as bitter pit, often associated with calcium deficiency. And sometimes the bitter pit starts from within the apples rather than the surface. So we use X-ray technique mainly for production rather than breeding. It can be useful for breeding as well. But in production, what happens, they store these apples for about a year, and in a few months, when they remove it from the storage facility, this bitter pit has developed. So we wanted to develop a technique where it can still be packed, and you scan it, and you will know whether the apples have this disorder or not. And when you look at the image processing aspects, there is similarity between both the projects. And of course, here again, the throughput is important, and we can achieve a throughput in 60 to 100 seconds. We are able to process all the X-ray CT data, which is much bigger. And of course, I say without intermediate images, one thing that we learn as a technologist or a data scientist is as you process the images, it's always better to once in a while check, is my algorithm correct? Because often what happens in the process, if there is a small error in image acquisition, say that the angle of the image acquisition changed, the data can be very noisy. So it's very important once in a while to see to make sure whatever you're seeing is what it should be. So So here I wanted to just give a sort of a brief piece of advice on different how to use image processing. The first one is know your sensors, right? So this is this is a multispectral image, and there are 3 bands. Usually a digital camera has R, G, and B, which is red, green, and blue. Here you have, instead of 1 channel, you have near infrared. Just looking at this image, you will be guessing that this is my near infrared, because plants reflect out near infrared much more than the colored, because they absorb more visible light for chlorophyll purposes, as well as they should reflect. But actually, this is the near infrared. So the first thing is you should know what spectral bands. Sometimes we have a student from plant science coming saying that they are using multispectral band camera. They don't know which band is near infrared. And sometimes it's very hard to figure out. So you should know your sensor is the first advice. A field of view and resolution is important. So we work with lentils. Probably that's the tiniest crop we have worked with. So if I'm flying a drone in a field of lentils, I should know like what's the altitude is best to give me the best resolution for that particular replication. And the third one is the reliability of the sensor. And I was at Phenome conference last week, and we had a speaker say the same thing. It can be the same company, it can be the same model, but they are making revisions to their version again and again. So if you're going to change your sensor, even if the model number is same, you may be changing what data you're acquiring. And sometimes we do that because we have a group of 8 breeding students that we are borrowing the sensors for throughout the season. There's a lot of demand. So we do have multiple sensors. And one of the advice we do is make sure we have a tag in every camera. You're using the same camera every time. Because if you're going to change the camera 1 and camera 2, probably, I don't know how, we do normalize all the images. Still, sometimes there may be some differences. The second aspect is compensation. We calibrate each of our, even RGB camera images, as well as visible infrared. I cannot insist more that calibration is very important. I'm not going to talk about it, but usually each light has something called as a color temperature. So you have to, between light sources, if you want to keep your data comparable, even cloudiness, solar radiation, it's very important to calibrate. And this is just an example. Here we are looking at LED light. LED light does not have all wavelengths equal, so they have differences. But once you calibrate it, you can almost get a natural color of the root. And the same way, thermal images also, we find that calibration is really improving the quality of the data. And the third aspect is the light quality and quantity. Especially if you are doing near infrared imaging, your light source needs infrared source. So it's a reflection that you're measuring. So your light should have a near infrared band to be able to reflect that. Imaging angle, time of the day, all those things play a role. Imaging protocol is very, very, very important if you need to automate the process. If I am able to process 25,000 images in 2 days, it is only possible if you keep the imaging protocol the same. So once in a while, based on your convenience, if you have changed things, it's going to affect your data quality. And image background is important. And sometimes this, I call it a cross-disciplinary communication. We had a prototype algorithm to do a seed size on chickpeas, and we used quite a uniform background. And a lentil student wanted to know. And I surely was expecting something like this, my lentil color would be. I did not realize they had so many different colors in lentils and even patterns within the lentil. And it was, And it's our mistake completely that this cross communication. So even if a plant science student is using a technology, they need to understand a little bit of what we are trying to do, so that both of them can understand each other's problems and get the best solution. So now, after this experience, we are sort of sitting with each student and establishing a protocol, because without this protocol, automation would not be possible. And finally, I talk about the oversaturation. It's very important. Sometimes you see the leaf surface shines, so it results in saturation. So if there is a saturated signal, there's no way you can correct it by image processing. I can still fix this problem up to a certain level. I can still find the relative intensities, edge of the seed to be able to still get a seed size from this image. But if my image is saturated, there's no way I can correct it. And this you have to make sure when you are in field while doing it. So basic steps of image processing. So when you take an RGB camera, there are different color space. So I said red, green, and blue. This is the standard color space. But of course, you can convert it into other color spaces like hue, saturation, and value. And there are many, many different types of color spaces that you have to choose. So you have to choose the one that best separates the background and your object. And in general, when you do a background subtraction, here is an eggplant with a background, and here is a field aerial image with a background. In general, the most commonly used process for background elimination is a histogram analysis. So if you do a histogram of this image, it's divided into two groups. The result for this image is this group. That's how we eliminate the background. This is the very standard method of doing it. Sometimes we use special methods, which I'll be talking about in a minute. So once you have your processed image, you have eliminated all the noise and all the background, you extract many different types of features, and you can have color, shape, texture features, and here I'm just naming a few. Just as an example, this was a study done by my graduate student, and our goal was phanomyces infection, and we had almost thousands of images. We had 360 lines, 5 reps, healthy as well as inoculated, and just a few steps, like she had this original image, she used hue color space to separate the background, and this is how, after eliminating the background, this is the image that she gets. She extracts about 87 features just based on RGB. And I say times 3 because the region of interest can be a shoot, can be a root, or shoot and root combined. So she extracts around 261 features. So it's a lot of features, and I often say sometimes too many features can confuse the machine learning algorithm, which I'll talk in a minute. So it's like within this room, if we all take if we take your blood samples or RBC and WBC level, there is a high variability in that. So you go to a doctor, you say, these are my symptoms, what do I have, right? Sometimes you can give them more, okay, my RBC level is this, WBC level. The algorithm is similar. The doctor may get confused. The same way the machine learning algorithms may also get confused. So it's very important to understand these features. So as a technologist, for me, I have to understand what is my breeder really rating in this. And of course, we did some correlation analysis, and we found that the root is more focused, and phanomyces is a root rot disease. It makes sense that the breeder is, although he or she is evaluating both roots and the shoots, they are focusing more on roots. And that's important as a technologist to understand the process. And I was talking about special methods. Instead, I wish that histogram thing always works. Sometimes it doesn't. And this is my original image. This is just by that histogram I talked about. And sometimes we use some special methods like k-means clustering. And if I have a lot of noise, I have to use it. And this is going to change my processing time. So if I can process 250 images in 20 minutes, instead of 20 minutes, it's going to take 4 hours if I'm going to incorporate a special method for background elimination. The same way here, if you see, again, the blue band is always higher than the green. We use the spectral differences. So we do blue minus green to say whether it's a potato or a background. So sometimes we have to do more than simple step. This will be still faster than this. So you have to, we are optimizing the process to see which is the best in terms of throughput. And again, here I'm talking about field. And sometimes you see this is a distortion. The plots over here are going to be much smaller and the plots at the end. And if I'm going to not correct it for distortion, it's going to again lead to a biased data. So we do distortion correction, and this is just an example of distortion correction. We do pretty well in automation. What we do is we pick the first point, last point, and equally divide it by the number of plots. So all these plots are divided so. So again, both this way as well as this way, because there are around 800 plots in this image. And if I'm going to manually go and pick out each and every plot, it's going to take forever. So there is a possibility of automating the entire process, and we do find some interesting and good results. Now I'm going to machine learning. So machine learning has been utilized quite a bit, especially in terms of big data processing. So broadly, they are classified into identification, classification, quantification, and prediction. And I will talk a little bit more detailed about it. Again, there is an overlap between each of these sessions as well. So when you talk about detection, I'm just interested whether the disease is present or absent. Okay, so it's present. It's Huang Long Bin. It's a citrus greening disease. And it's present or absent. It can be nutrient deficiency that looks very similar to Huang Long Bin. It's present or absent. If it's a healthy, it should ideally, if the machine learning algorithm is working properly, it's still absent. So there are only two categories, present or absent, when we talk about the detection. When it comes to the classification, I have different categories. I have healthy, Huang Long Bin, and citrus canker, another types of diseases. So when the machine learning algorithm is classifying different categories, then I say, okay, this is healthy, Huang Long Bin, canker. I can have other categories. I have nutrient deficiency. I can say nitrogen deficiency. I can say potassium deficiency. You can give as many classes. But this was one of the most interesting disease I ever worked with, because the symptoms are never the same. So if you grow them in the greenhouse, the symptoms are different. If you grow them in the field, they are different. And sometimes the symptoms are pretty hidden inside the canopy. So it was very challenging. Okay. When it comes to the quantification, so this is an example of hail damage in potatoes. And this, I say it's a 0%, 33%, 66%, and 99%. So though this study does not summarize the machine learning aspect, but it's just to show you as an example, it's categorizing, and it's no more qualitative. It's a little more quantitative, right? I'm just saying little more quantitative, because the best thing that you can ever get through the machine learning is the prediction. Here I have water use efficiency, transpiration rates, tomatal conductance, and I am predicting with my visible near-infrared spectra what is the transpiration rate, what is the water use efficiency. And this I would say that's the most challenging aspect in a machine learning algorithm to be able to predict. The reason being, biologically, I can say 10.1 is same as 10.5, right? But when a machine learning algorithm is looking at 10 and 10.5, it may perceive it as different if the spectra between the two is different. So it's very different when a machine learning looks at it versus we look at it, right? So what is machine learning? So machine learning is nothing but it's learning patterns, similarities, dissimilarities from the data. And I think it's all of us have the same thing, right? When you're sending an undergraduate student or graduate student to phenotype, they are learning patterns. Okay, how do I rate this disease? How do I rate water use efficiency or senescence, right? So machine learning often deals with big data, so it's much more easier to handle big data sets with machine learning algorithms. And I do not know anything about bioinformatics or genomics, but I come across the same terms. The algorithm I'm using to handle phenomic data, sometimes it's very similar. So the methods are very similar, just that we are using it for different applications. So, and machine learning is used, sorry about that, in practical sense, like for potato industry, they scan each and every chip, right? So if they see a browned chip, they are going to, the machine learning algorithms that is connected with the camera, it's going to make that decision, this chip is not of good quality, it's not going to go to packaging. So it is used many terms in practical sense. Another good example is face recognition, right? So, or fingerprint, right? So if do I have a criminal record, right? How are they going to do without the machine learning? They are using machine learning approaches to be able to do that in a rapid throughput manner. And many tools, like nowadays, it's very much easy. MATLAB have their own machine learning algorithms. Previously, when I was a graduate student or a postdoc, I used to write 10-line code to do a 1-line code that is now, right? So now I need to develop this complex code to be able to do the machine learning. Now it's inbuilt in many of the software. And just last week, I learned that PlanCV is starting to incorporate some of those machine learning approaches as well. Again, training is important. Just like graduate students or undergraduate students going to phenotype, training is very important for machine learning approaches, right? What is a large data set? So the larger the data set, the better is your prediction or classification, whatever you may say it, right? Whatever is your application. But what is a large data set? How do you come up with large data set, right? What is the magic number? There is no magic number. It depends on the application, right? If your plans are showing high variability, you need maybe thousands and thousands of data points for the machine learning approaches to perform the same thing. If it's a simple disorder that's very consistent and you do not have much higher variability, your data size can be relative, maybe hundreds. 50s and hundreds would make sense. So it depends on the variability of plants. And I think even in phenomics has a very high potential in picking out the variation within a variety, right? So sometimes you do plant height, right? The maximum number I have heard for a plant height is 3. Hardly, they will take a yardstick, at least our wheat breeders take a yardstick and measure 3 different plants. The phenomics, if you use a LiDAR, you may be able to capture much more variability within a variety. And I think that's the advantage of using the phenomics, at looking at variation within the varieties. And so this is just an example of what I spoke. So you have a training data set, you have a test data set, and you have the raw data. So in training data set, you are going to say, this is my healthy plants this is the way it looks like. This is my disease plan. This is the way it looks like. Now you go and I still give a test data set and I know how it performs. This is for model development. After that, ultimately, what I want to do is I should have an unknown data set, and still my algorithm is able to perform well. That's the real challenge, right? We never have 2 seasons the same, right? I wish it was. If we had, then probably our algorithm is going to perform much better than we expect. And again, there are 2 more terms I wanted to introduce. One is a supervised learning. The one I just talked in previous example was an example of a supervised learning. Say that this is how my healthy looks like. This is how my disease looks like. Go and find out which ones are healthy and disease. That's a supervised. I'm telling it, I'm giving it a label. Sometimes we use unsupervised. Say this is all the data. I'm not going to tell you what's healthy. I'm not going to tell you what's disease. You figure out, are there groups within this data set? And here is an example that we found that 3 different groups existed within the data set. We are not telling anything that this is healthy. That's an unsupervised learning that can be also utilized for detection. I'm not going to go much over it, but there are many different models that can be used. And we can process. I'm just going for the time, I'm just going to skip some of these slides. Here is an example where I mainly talk about the apple breeding programs, where, yes, the disease is an important factor. Graft compatibility is another important factor. Tree architecture is very important factor. Especially in Washington, all our architectures have a structured structure just for the ease in harvesting and other aspects. In that case, it's important to adjust the variety for tree architecture. And of course, in tree fruits, we usually have overproduction. Sometimes some orchards are not harvested because there's no place to store, right? But when it comes to quality, they are constantly trying to improve quality. McDonald's did not have apples 10, 15 years back, but now we are moving towards healthier options. So quality, you should be able to cut your apple, keep it in the table for a few minutes and not avoid browning. That's one of their objectives, right? When you look at the rose breed guidance for how you should phenotype, you see these are the 8 guidances. And in that, you say that throughput is an important one because there's a limited window in which you have to phenotype, right? And personal fatigue, repetitively doing the same thing can result in loss of reliability, morals, as well as personal sometimes, right? And if you really see high throughput phenotyping can assist in at least 5 of those 8 points. So it has a lot of potential. And I'm just going to quickly give an example on fire blight resistance in apples. This is an important disease, can result in a loss, a significant loss. It's 100 million for maintenance, for avoidance as well. The Wenatchee, Washington is called the apple capital of the world. So we have our apple breeder, Dr. Kate Evans. They are doing phenotyping and they have already done 2 seasons. They are not going to go for 3rd season because it's pretty labor intensive and expensive as well. So what we did was we used many different types. We used visible near-infrared spectroradiometer to get and machine learning approaches. We did gator-based imaging and we did aerial imaging. And when you inoculate one shoot in the entire tree, you're not expecting changes in the canopy level, right? You're just measuring that one branch that's inoculated. But still we wanted to see aerial because to see if that is inducing some stress in the entire tree that we can still be able to see. We were ambitious a little bit, you can say that. Sometimes we get a good match with the ground rating, sometimes we don't. So we completely relied on the visible near-infrared spectroradiometer. Of course, what we did is imaging and just for the time's sake, I'm just going to say often your symptoms, which is senescing, is similar to the other ones, and you have to do some special transformation to be able to identify those symptoms. We get good classification accuracy. We were fortunate, though 2016 and 17 were completely different, we still got similar results. And these are some of the vegetation indices that we have picked out of those, because you don't want a breeder to collect again a big data set, right? You want them to quickly be able to evaluate. This is again post-harvest side. With that, I would just like to summarize by saying that hyperspectral with machine learning approaches have a lot of potential. Phenomics is an integrated approach. We are learning a lot. We as technologists or data scientists are learning a lot. And it's important that the breeder also understands the limitations and benefits so that you can optimize a process. And image processing, machine learning can learn to lead to a biased data. For example, sometimes you see 100% accuracy. I teach instrumentation and measurements. For me, there is nothing called as 100% accuracy. No sensor is 100% accurate. It can be 99.999, but never 100%, right? So it can lead, you can modify it, and sometimes we have our graduate system, students trying to see what they want to see. You can make it happen. So it's very important that you do an objective evaluation across season, across different locations. That's very important. With that, I would like to acknowledge my graduate students and a bunch of very, very, very good collaborators. Most of them are breeders from USDA and ARS as well as WSU. And whenever I have a new experiment that I want to evaluate, they are doing a lot of volunteering for planting for me, because I'm very bad at it and I can kill plants easily. So they volunteer a lot and are excited about the phenomics research. With that, that's our beautiful Palouse. I would like to thank you. And sorry, I had to skip few slides, but I'll be happy to answer questions.
Thank you, Dr. Sankaran, for your presentation. If anyone in the audience has a question for Dr. Sankaran, we will now begin a short question and answer session. If you please raise your hand, and one of our volunteers will come up to you with a microphone. For those watching online via webinar, if you would like to ask a question, please post them on the chat box. Dr. Murray.
One of the things you started off with was saying how important it was to correct things to a reference for the image, and then to standardize cameras. But I guess one of the questions I have is, in a plant breeding program where we're really trying to make comparisons between things within the same picture, how important is that when we're looking at relevant differences rather than absolute differences?
That's a very good question. And like, for example, in aerial image, right? So I have some 400 plots. If I'm capturing one image of those 400 plots and only making comparison between varieties the same day, the same time points, then probably you don't need a calibration. But we have never done that. We usually have multiple time points and multiple locations. So we think it's important to do calibration. And if you publish any sensor journal, probably they will not accept it if you do not calibrate. It's a standard method. There are also easier ways of doing, which I haven't told over here. Sometimes we use this panel, the reflectance panels. Nowadays, we get this light sensors, and the camera itself can automatically adjust based on incident light. So things are getting better now. We don't need a reference panel in the field. So things are improving. So I think, yeah, across, yes, sometimes previously we were doing that with temperatures because temperature was much difficult to calibrate than a reflectance. But nowadays we find that if we do calibrate, we are getting better data, so better correlations.
Any more questions? Dr. Jean.
Yeah. My question is that I saw you use the cross-validation data, right? Did you try independent validation?
So we did not use cross-validation. Most of our study, but I wanted to talk, there are two types of validating the data. One is independent validation
Yes.
with test data sets if you have big data. But if your data set is very small, you can still do a cross-validation, but it's cross-validation can be biased. So I just wanted to talk about two different ways in which we can test, but most of our data is independent test data sets.
Oh, independent. So there's a different accuracy, prediction accuracy different between two validation?
Yes, it will be different in most cases. So independent testing, usually the accuracies are lower than cross-validation. So an example of the difference, just for the audience sake, if you have 10 data points in an independent evaluation, I'm going to use 8 data points to develop my model and 2 data points to say that, okay, based on this model that I have trained, is it able to predict correctly? I'm saying 2, but it's 20% or 80%, or ratio may differ. When it's cross-validation, I have this 10 points. I use 9 points, predict 1. 9 different points, predict 1. Like that, I may do 10-fold, I may do 10 times, and then say, okay, so it's almost like it can lead to improved modeling, but the accuracies will ultimately increase because you're using the same data set again and again to develop your model.
Another thing, that means if you did one population, breeding population, you can use another population without a problem. Is that right? If your model developer use
yes.
one population, let's say,
I would,
and you can use the other population.
Within the panel, if you change the panel, your results are going to differ.
And another thing, so you did this year, can you the data use next year for the prediction?
That's our goal. That's our goal.
Yeah, because that's important for breeding.
That's within the panel. So in dry bean, we had, I don't remember the top of the names, Durango population or something. And we had, if it's the same panel across different season, multiple locations, the sensing can work. But if your panel is changing, sometimes the leaf structure also changes, so it may affect your results.
Any more questions for Dr. Sankaran? We have one in the front of the room.
So in the instances where you're using this to look at whether or not a disease is present, so in citrus greening and the fire blight, what is the threshold of detection? I guess I'm asking, like, can you see the symptoms before you can detect it using your...
Yes, and that brings a big difference between phenomics and precision agriculture. In precision agriculture, we are interested in early detection. So in that particular study, though a breeder would ideally wait for 6 weeks for maximum development and only evaluate at the end of season, we did data collection beyond that time point to make sure how early can we detect. And at least in that study, we see that we can find those differences much before the final stage of raking. So there's something upon inoculation, something is happening to the plant that we can pick out much earlier.
Do we have any more questions?
Yeah, you talk about the calibration before you use the sensor to collect the data. How do you calibrate the sensor? Do you use the specific traits before you use the calibration? Because in practice, in the field, you have several symptoms. For example, there has the disease, nutrition deficient. So how do you, if you calibrate it to the specific traits, but in actually you have several symptoms in the field, how do you deal with this problem?
That's a very good question. So I think you're a little confused on the two aspects. One is the calibration, right? So the calibration is to make sure the sensor is working correct, right? So that's a different that we do with all our sensors, make sure the sensor is working correct. The second aspect that you mentioned is you have a lot of things going on in the field. And when I got this position around 4.5 years back, I thought it was exciting opportunity because in precision ag, that's important where you have nutrient deficiency. Sometimes you have overspraying, so sometimes you have drought. It's very challenging given the opportunities. For breeding, I was excited because you have a very controlled experiments. If you're looking for disease resistance, you're making sure you have enough nutrients, enough irrigation if it's an irrigated, and only varying one parameter at a time, sometimes two parameters at most. So I think for breeding, it still makes sense because that variability is much more controlled. Sometimes I see weeds in the plots and it can create noise unless that weed is very different looking that I can still remove with images. But I think in practical sense on can I detect a stress? I would say probably even if based on drone based images, it can tell you something is wrong with this tree. It's sick. That's all it can tell you. What type of sickness, you have to go closer and do a little more evaluation. Still, it improves efficiency because instead of scouting the entire orchard, you're going to just focus on few points that are not right. And your sampling may also be reduced. Instead of sampling the entire group, you may just sample a few trees to identify a particular condition.
Do we have any more questions for Dr. Sankaran? If so, let's thank Dr. Sankaran once again. On behalf of the organizing committee, I encourage you to share your experiences at our event on social media platforms and tag us on all these posts. We would like to thank our sponsors once more before we adjourn for a 15-minute break. Please help yourself to coffee that is in the back of the room, and we will begin with our next speaker promptly at 10:15. For those of you who are looking for restrooms, they're at the end of the hallway on the left side.